1 2 3 4 Media Bio.
This is the part 5 of the Human Brain series. Please do watch all the previous parts before going ahead with this video. Now we will look at the physiology of the brain. So our first topic is ventricles, not of the heart, but of the brain. Unlike heart, the ventricles of the brain is somewhat in this way: lateral ventricle, third ventricle, fourth ventricle, and subarachnoid space. Let's look into the detail. The brain has hollow fluid-filled cavities called ventricles. There are two ventricles, deep within the cerebral hemispheres called the lateral ventricles. They both connect with the third ventricle through a separate opening called the foramen of Monro. The third ventricle connects with the fourth ventricle through a long narrow tube called the aqueduct of Sylvius. Please note how the foramen of Monro and the aqueduct of Sylvius is written. That's how you are supposed to write and maintain the upper and lower case. From the fourth ventricle, CSF, cerebrospinal fluid, flows into the subarachnoid space, and finally into the brain. CSF is recycled, or absorbed, by special structures in the superior sagittal sinus, called the arachnoid villi. The previous points, along with these 2 points, will become clearer when we will go back to the previous diagram, and I will add 2 more structures to it: foramen of Monro and the aqueduct of Sylvius. Well, take a proper look into the diagram and try to relate it to the points that we studied till now. You may pause this video here, or may go back a few seconds. So pause here now. So hoping that you understood the ventricles of the brain, let's study about cerebrospinal fluid. Inside the ventricles is a ribbon-like structure called the choroid plexus that makes clear colorless cerebrospinal fluid, CSF. CSF flows within and around the brain and spinal cord to help to cushion it from injury. This circulating fluid is constantly being absorbed and replenished. That's good. CSF is produced inside the ventricles, deep within the brain. CSF fluid circulates inside the brain and spinal cord, and then outside to the subarachnoid space. Continuing. A balance is maintained between the amount of CSF that is absorbed and the amount that is produced. A disruption or blockage in the system can cause a buildup of CSF, which can cause enlargement of the ventricles, or hydrocephalus, or cause a collection of fluid in the spinal cord. You can call this syringomyelia. So hydrocephalus and syringomyelia are the 2 kinds of diseases due to excess of CSF. Now we will study about the cranial nerves. The brain communicates with the body through the spinal cord and 12 pairs of cranial nerves. Cranial nerves originate from the brainstem, exit the skull through holes called foramina, and travel to the parts of the body where they innervate. Well, this point is important, so reading it again: cranial nerves originate from the brainstem, exit the skull through holes called foramina, and travel to the parts of the body where they innervate. 10 out of the 12 pairs of cranial nerves that control hearing, eye movement, facial sensations, taste, swallowing and movement of the face, neck, shoulder and tongue muscles, originate in the brainstem. The cranial nerves for smell and vision originate in the cerebrum. So as we got different cranial nerves, performing different functions, we will look into the detail, which is present in this table. Let's start from cranial nerve number 1. Its name is olfactory, and it performs the function of smell. Next is number 2. Its name is optic, and performs the function of sight. Number 3 is oculomotor, which is used during moving the eyes or pupils. Number 4 is trochlear, which also moves the eyes or pupils. Number 5 is trigeminal, used during kind of face sensation. Number 6 is abducens, which moves the eyes. Number 7 is the facial, which moves the face, and also helps to salivate. Cranial nerve number 8 is vestibulocochlear, which is used during hearing, and maintaining balance of the body. Number 9 is glossopharyngeal, which is used to taste food, and swallow food. Number 10 is vagus, which is used to maintain heart rate, and digestion. Number 11 is accessory, which moves the head. And finally we got number 12, which is the hypoglossal, which moves the tongue. So these are the 12 pairs of cranial nerves. I would recommend you to take a screenshot of this slide. Here's the diagram of the brain, which shows all the cranial nerves. Pause this video here, and take a moment to look at the locations of all the cranial nerves. Time for meninges. The brain and spinal cord are covered and protected by 3 layers of tissue, called meninges. From the outermost layer, starting from top, they are the dura mater, arachnoid mater, and pia mater. Here's the 3-layered tissue meninges. It shows the dura mater, brain, skull, arachnoid mater, and the pia mater. Dura mater. It is a strong and thick membrane that closely lines the inside of the skull. Its 2 layers, the periosteal and meningeal dura, are fused and separate only to form venous sinuses. The dura creates little folds or compartments. There are 2 special dural folds, the falx and the tentorium. The falx separates the right and left hemispheres of the brain, and the tentorium separates the cerebrum from the cerebellum. Arachnoid mater. It is a thin, web-like membrane that covers the entire brain. The arachnoid is made of elastic tissue. The space between the dura and arachnoid membranes is called the subdural space. The final layer of the meninges is the pia mater. It is attached to the surface of the brain, following its folds and grooves. The pia mater has many blood vessels that reach deep into the brain. The space between the arachnoid and pia is called the subarachnoid space. It is here where the cerebrospinal fluid bathes and cushions the brain. Alright, here we end up the 3 layers of the meninges, dura mater, arachnoid mater, and the pia mater. Now let's study about the blood supply to the brain. Blood is carried to the brain by 2 paired arteries, the internal carotid arteries, and the vertebral arteries. The internal carotid arteries supply most of the cerebrum. The vertebral arteries supply the cerebellum, brainstem, and the underside of the cerebrum. After passing through the skull, the right and left vertebral arteries join together to form the basilar artery. The basilar artery and the internal carotid arteries communicate with each other at the base of the brain, called the circle of Willis. Continuing. The communication between the internal carotid and vertebral basilar systems is an important safety feature of the brain. If one of the major vessels becomes blocked, it is possible for collateral blood flow to come across the circle of Willis and prevent brain damage. Wow, this is a lifesaver. Look at all the blood vessels of the brain. Take a moment to study the diagram properly. Cell organization in the brain. There are 2 types of cells in the brain: nerve cell, or neuron, and the glia cell. Let's look into detail. Glia, also called glial cells or neuroglia, are non-neuronal cells in the central nervous system, brain and spinal cord, and the peripheral nervous system, that do not produce electrical impulses. Glia cells outnumber neurons in the mammalian central nervous system, and are key to maintaining tissue homeostasis. There are 4 kinds of glia cells: astroglia, oligodendroglia cells, ependymal cells, and the microglia. Let's look into their detail. Astroglia, or astrocytes. They regulate the blood-brain barrier, allowing nutrients and molecules to interact with neurons. They control homeostasis, neuronal defense and repair, scar formation, and also affect electrical impulses. Next, oligodendroglial cells. These create a fatty substance called myelin, that insulates axons, allowing electrical messages to travel faster. Next, ependymal cells. These line the ventricles, and secrete cerebrospinal fluid, CSF. Finally, microglia. These are the brain's immune cells, protecting it from invaders and cleaning up debris. They also cut out synapses. Wow, all these 4 glia cells are thus important. Alright, now check this diagram. It shows all the glial cells, and their structures. You may pause the video here, to have a good look at the diagram. So finally we end up the part 5 of the human brain. See you soon in the part 6, and hey, please comment down below, anything you may like. And please subscribe to our channel 1234 Media Bio, to keep information like this, in your brain.